Just like any other existential questions, as curious creatures humans are, the age of Earth has always been a mystery to scientists. Well, up till this day, scientists are still investigating proofs to support current theories. One of the evidences that helps scientists find proof to assist theories related to the age of Earth and understanding the history of our existence is by determining whether or not life exists on other planets, which is a mystery until this day. One of the techniques scientists have used over time to determine how old Earth is by studying changes of time on rock. Imagine yourself making a layered cake. The lower layer made first has been there for the longest amount of time. Layers of sediment, rocks, and sand deposit in the same way. These layers eventually harden into rocks. Just like leaving the cake, the layers at the bottom most likely start drying first.
Using this method to determine relative age, which is science of determining the relative order of past events without necessarily determining their absolute age. In geology, rock or superficial deposits, fossil and lithologies can be used to correlate on a stratigraphic column with an event.
The early Earth was very different from our Earth today. Gases created a new type of atmosphere for Earth. Consequently, the Earth was very hot, evaporating the liquid water into the atmosphere. However, as the Earth cooled, gravity-trapped water vapor condensed, fell as rain, and did not boil away but remained impounded in pools that became lakes and oceans. It was also believed that tectonic activity caused many volcanic eruptions at that time. From present-day volcanoes, we know that when they erupt, they release carbon dioxide, nitrogen, and a host of non-oxygen gases. In addition, with no protecting atmosphere, the Earth was constantly bombarded with meteorites and other space debris still in circulation from the Big Bang.
Even though scientists don't agree on how life on Earth began, it is agreed upon that living things require organic molecules as building blocks in order to survive. As we know, water is essential for living things, and this led researchers to believe that life actually began on the oceans. Scientists began to experiment this to try and prove that abiotic synthesis of simple organic molecules could have occurred in the early Earth oceans. One of the most famous experiments concerning this hypothesis was that of Stanley Miller and Miller-Urey in 1953. Their hypothesis stated that organic chemical compounds can be generated under conditions similar to those that existed in the atmosphere of primitive Earth. The experiment went like this. First, they heated a solution of simple chemicals to produce an atmosphere of methane, ammonia, hydrogen, and water vapor. Next, the vapor traveled to a large flask where electric sparks, stimulating lighting, provided energy for synthesis of new compounds. Then, a condenser cools down the atmospheric gases to a rain containing new compounds, which is then collected in an ocean. Finally, they collected the condensed rain and analyzed it. The results show that the reactions in the condensed liquid eventually formed organic chemical compounds, including amino acids, and so the conclusion that they reached was that the chemical building blocks of life could have been generated in the probable atmosphere of early Earth. Now you may be asking, but how do complex organic molecules form from simple molecules? To answer this, first you need to know what monomers and polymers are. Monomers are the basic building blocks that when combined form polymers, and for every 2 monomers that join, 1 molecule of water is released. Now, how does that happen? One of the most common ways is by removing one OH group from one monomer and an H group from the other, thus leaving space for a covalent bond to be formed between the 2 monomers. Now, the atoms that will be removed, the 2 hydrogen and oxygen, they will combine to form 1 water molecule of H2O. This process is called a dehydration reaction because we are taking the water out of the polymer. Scientists hypothesize that countless of these reactions have been occurring over time, and complex organic molecules could have accumulated in the primordial soup.
But how do we go from primordial soup to protocells? Once these biological molecules were formed, they needed to become integrated with one another to form living organisms. But to do so, they needed boundaries to separate them from the outside environment. In the 1920s, scientists discovered protobionts or protocells. Protobionts are small membrane-borne structures that assemble in water as the water temperature changes. They contain organic molecules and often have an extra lipid or protein coating with their simple outer membrane. This early cells-like structure could have protected organic molecules from the outside environment. According to this hypothesis, small organic molecules, including amino acid and nucleotides, were synthesized, combined to form polymers. Once boundaries were established between the external and internal environment, unique chemical reaction could occur. Researchers have hypothesized that some RNA, such as ribozymes, behaved as an enzyme like a catalyst, capable of catalyzing many metabolic reactions.
When placed in water cool enough for base pairing, but without enough free nucleotides for replication, chains will fold up and base pair with themselves. The end result is a complex shape with certain sticky bases pointing outward because they weren't able to find partners. These sticky outward-facing bases can cause unique chemical reactions by interacting with other molecules in their environment. A folded chain of RNA capable of guiding a specific chemical reaction is what we call a ribozyme. Some ribozymes break certain molecules apart, others join certain molecules together. A ribozyme's specific function is determined by its specific shape, and its shape is determined by its sequence. If a mutation changes a ribozyme's sequence, the shape can be modified, and so can its function. When ribozymes were first discovered, scientists wondered how difficult it would be for random chains of RNA to evolve legitimate survival functions.
The name ribozyme comes from the ribonucleic acid RNA and an enzyme. Ribozymes are able to synthesize copies of short pieces of RNA when supplied with nucleotide building blocks. RNA in today's world. Today, DNA and RNA is considered the major genetic molecule of cells. So at some point during the evolution of cells, DNA took over the role of RNA because a double-stranded DNA is more stable and easier to replicate.
The evolution of unicellular life. Two of the main events that drove evolution on early Earth were the incorporation of DNA as a stable genetic material of organisms and the ever-changing atmosphere. At the beginning, Earth's atmosphere contained no oxygen. The earliest prokaryotes were therefore anaerobic and thus carried out metabolic reactions without oxygen. Scientists think that the Earth formed around 4.5 billion years ago. The early atmosphere contained no free oxygen, no ozone, and plenty of gases that would be toxic to most life forms today. As the planet began to change, it caused an increase in volcanic activity. The volcanoes outgassed materials that formed an atmosphere. It is thought that one of the gases released was water vapor. As the planet and atmosphere cooled, condensation occurred, and large bodies of surface water formed. Molecules in the atmosphere somehow combined into complex compounds. Miller-Urey's experiment shows that this would have been possible. Once complex organic molecules were contained, prokaryotic cells began to form. Prokaryotic cells that were able to perform photosynthesis produced the first free oxygen and eventually changed the composition of atmosphere to one that contains 21% oxygen. But how did the evolution of eukaryotes and multicellular life occur? Well, prokaryotes rule the planet for a large part of Earth's history, but your cells and the cells of other complex organisms are eukaryotic. The first eukaryotic cells are believed to be the result of a predator-prey relationship in Earth's oceans roughly 1.5 billion years ago. Scientists believe that endosymbiosis may be the reason modern-day eukaryotes have organelles. The following video will give you a more clear understanding of how unicellular eukaryotes became multicellular.
Each prokaryotic cell has a stiff cell wall, which protects the cell from damage. It also contains genetic material, loosely arranged in the cytoplasm. More complexity in larger sizes could be possible only if the outer layer was lost, resulting in a flexible cell membrane. This allowed for folding in of the membrane, which increased the surface area for absorption of nutrients and release of wastes. The infolding also resulted in the formation of membranes around different sites in the cell, one of which housed the DNA. Meanwhile, internal support was developing in the way of the cytoskeleton. The flexible membrane also allowed the cell to engulf larger molecules and even other smaller cells. This resulted in the entrapment of prokaryotic cells that would become mitochondria and chloroplasts in modern eukaryotic cells.